Welcome to the Dinosaur Soft Tissue Research Institute Master Class Laboratory. This is a lab that we've set up with several workstations. As you can see, we can accommodate up to 8 students in this laboratory, and pretty soon students are going to start coming to get more specialized training on how to collect and handle dinosaur soft tissues, and we'll talk more about that later. But I'm Dr. Mark from DSTRI.org, and we have not done an update for quite some time, so we thought it was important to provide an update to all the folks out there on what we have been up to, and it is a lot. So this is going to be a bit of a long video, but we've divided it up into sections to make it easier for you to start and stop and come back to where you were and watch each segment differently at a different time if you need to. So we have a lot to cover. Section number 1 is going to deal with our publications for 2024. We have 3 excellent world first publications to share with you so far this year. Section number 2 will talk about our students. We have students in Washington State here, in Oregon, in Portland, Oregon, and also in Texas, and they've been very active, so we're anxious to share what our students have been doing. Section number 3 will discuss our wonderful research associates who have been doing some excellent work along with DSTRI.org. In fact, they have exciting nematode worm results to report. So, and also our other research directions at this time of the year, since we've just come back from digs. Section number 4 will be about our digs. We went to Wyoming this year. We also went back to Montana, and we collected some very interesting specimens, so we have a lot to share about that. Section number 5 will deal with the Microscopy Society of America annual meeting, Microscopy and Microanalysis, in Cleveland this summer, where we attended and presented our work. One of our students also presented, and so we're anxious to share how he did and the offers that he's already gotten. So it's really exciting to see how our students are growing and developing into a wonderful career. And section number 6 will be about your student, how your student can take advantage of the opportunities that the Dinosaur Soft Tissue Research Institute offers. We publish in leading microscopy journals. We have lots of research lines that we're anxious to follow. And so we need students to come and learn these techniques and these skills. They'll learn about microscopy, which is a wonderful career opportunity. And so we're anxious to share some of the opportunities that we have for students who want a career in science but need to gain the skills to do that. And so I've had a wonderful career in microscopy, almost 50 years, and we're anxious to share that with other students who can grow into this amazing field of science and earn a very lucrative living and have a lot of fun doing this. So stay tuned for all the things that we have to share. Section 1 is a review of publications that we had this year in 2024. As you know, DSTRI.org is one of the few organizations on the planet that is routinely collecting and publishing on novel dinosaur soft tissue discoveries. And this year we've had 3 publications in 2024. Our first publication was published in Microscopy Today, one of the Microscopy Society of America journals, published by Oxford University Press. Our other 2 papers were published in Microscopy and Microanalysis, which we'll talk about in a minute. But this is a very significant world-first discovery of nematodes in many individuals of dinosaurs that we've studied. We collected 11 different specimens from 9 different organisms, Nanotyrannus, Edmontosaurus, and Triceratops, different individuals in Montana, and all of them had large infestations of nematodes in the bones. Here's an example of a living nematode that was caught by the fruit trap method. You can try this at home. Slice off a piece of apple, put it in a small depression in the soil, water it, and come back in 24 hours. Then you can place the fruit slice in a Bowerman funnel and wash the nematode out and down the funnel into a collection container. Then you can put it on a slide, but you'll need a microscope to examine it. These are the kind of nematodes that we're talking about in these bones in Montana. Now, we routinely put our bones in a preservative at the site, so anything inside the bone gets preserved pretty much right away. This fixative works to our advantage, as one time we collected an entire nematode right out of our decalcification solution, so fixation seems to work best right at the site. And so we have found all of these once living nematodes which were preserved inside these bones when we put them in the fixative formaldehyde, or as we call it, formalin. This is a world first discovery. In fact, others have seen these structures in the bones and have mislabeled them. They've called them intravascular parasites, which are much smaller than the nematodes that we're finding. But the sizes of the nematodes that we're finding in these bones approximate what the size of living nematodes are. And so we published a whole series of these infestations, a world first publication in these dinosaur bones. Our second publication was published and presented actually at the Microscopy Society of America meeting and published in the Microscopy and Microanalysis Journal. It was a joint paper between myself and our intern, 16-year-old Jonas A. Cruz from El Paso, Texas. And we published on astonishing soft tissue permanence in surface collected Triceratops shards from Hell Creek, Montana. The point of this paper was that we went out and collected surface shards. We didn't dig for these specimens, they were on the surface. And so we weren't sure what we were going to find, but we found plenty of soft tissues in them, and we presented this actually at the Microscopy Society of America meeting. We also published a second publication in Microscopy and Microanalysis this summer, but this time examining the clots, the blood clots that we find throughout the vascular canals in these bones. And so we noticed that the clots were disrupted, not significantly, but enough to show that they've been on the surface for a while. We're very proud of our publication history, and this is something that we offer our students, as we did Jonas and others who are working with us. You have an opportunity to join DSTRI as a research associate or as an intern and work on these bones that we're collecting in Montana and make world first discoveries and get them published. So this is an opportunity for students to learn these skills, to learn what it takes to do a career in science using microscopy, and to publish at the highest levels, even before you graduate high school. So we think that's a really neat opportunity. Section 2, our students. We're very excited to have the caliber of students that we have, and it's exciting to see our student cohort grow. One of the things that we're very excited about is that there are so many opportunities for them that are opening up through the Microscopy Society of America. The local area societies are local extensions of the Microscopy Society around the country, and they are opportunities for students to present their work with DSTRI. So their research results can be put into a PowerPoint or a poster presentation, and these students can present at the local area societies with other students and get relationships with students there, and also make contacts with folks in their local area who can help them in their career. And so we're seeing these things happen, and we really have to brag about one of our students, Jonas Cruz, who's from El Paso, Texas. He's a high school student there, 16 years of age. And Jonas has been on a wonderful track over the last 2 years with DSTRI. He started out in one of our hands-on microscope labs that we've offered around the country. And then he came back and actually served as an adult volunteer, even though he was 15 at the time. But we were so impressed with his capabilities that we pressed him into service right away, and he did really well in assisting with the teaching. Since then, he's also presented at the local high school, Socorro High School in El Paso, and he taught actually 7 or 8 periods, and the students were really jazzed about his presentations. But he won our Most Improved Student Award in the spring, showing great promise with his research results. And he actually won a $750 travel award, a cash award, that allowed him to go to the Microscopy Society of America annual meeting, Microscopy and Microanalysis in Cleveland. We worked hard to get his paper submitted early before the meeting, and his paper was accepted. It was converted into a poster which he presented, and we'll talk more about his presentation when we discuss Microscopy Site of America and the many opportunities that are there. But he's now published at the highest level in the Microscopy and Microanalysis Journal. And so we're very, very excited. He's made some contacts that we'll talk about. He's also now a member of the Microscopy Site of America. So we are really, really very encouraged and excited and proud of Jonas for his work. Right now he's deep in research. He's working on decalcifying bones of Nanotyrannus lancensis, which were collected in Jordan, Montana. And so he's getting some very interesting results so far with that project. We have other students here in Washington and in Portland, Oregon. We're very excited about the students in Oregon because they are right next to a local area society there in Portland. And so they have an opportunity to interact with the PhDs there. They actually went on a field trip to one of the national cryo-electron microscopy laboratories, which is looking at proteins. They actually freeze proteins and look at them under transmission electron microscopy. And so there was a wonderful opportunity for the students to get in to this lab and to meet some of these professors. And so we're excited about their work, which is continuing. They're working with bones from Montana now and learning to decalcify those. And so there's a lot of opportunities for students, for our students, to get into these facilities, to meet with some of these professors, to make contacts long before their college experience. And so we're very excited to make those reports for the progress that we've made in 2024. Section 3 discusses our research activities in 2024 and our research directions. Because of the findings that we are making, this opens new avenues for research, and so this is why we need student involvement to help us do this decalcification, to look at the thin sections we're making from these bones, examine them for the clots and nerves and other things, the characteristics of these clots. And so we're making great progress on all these fronts. We really do want to brag about one of our research associates, Kirsten Peterson, who lives here in Washington. She has a Master of Science degree. She's a super duper mom who is rocking it with her husband and her children. She's actually teaching chemistry classes for homeschool families in her area here in Washington. So Kirsten designed a wild nematode capture experiment, which she had implemented on our dig in Montana in Glendive this May. And she did have success. We had success with this method. It was a very laborious method. It involved charting out areas and flagging them that soil samples were going to be collected from. She designed a modified Barrowman collection funnel for the field work. Her husband, who's an engineer, actually made it. It's a very durable and field-ready device that did give us success. One nematode was captured from this soil method. And so samples of soil were collected, cataloged, and they were processed through these field modified Bowerman funnels, which worked flawlessly. And one nematode was actually collected with this method. It's kind of a novel method. It has to be worked out, but we're just so proud of Kirsten for designing this, for designing the tools that were necessary to pull this off. And we had results, and we'll talk more about that and talk about our Montana dig and our Wyoming dig. But we are so proud of our research associates. Also, Keith Holcomb is actively decalcifying bones and collecting soft cells, nerves, and vessels from these bones. Section 4, our summer digs in Wyoming and Montana. Our first dig this summer was in Wyoming at the Hansen Ranch. This is a world famous, highly renowned ranch with an amazing collection of bones, a very dense collection of bones. This is run by the Southwestern Adventist University and Southern Adventist University on the East Coast. Southwestern, of course, is in Texas. And we'd like to acknowledge and thank Drs. Keith Snyder and Art Chadwick and also Dr. Jared Wood. They're running an amazing dig there with a lot of student involvement. They had something like 100 students digging at this dig this summer. To date, they've collected 30,000 bones, and they're in pristine condition. They don't show signs of abrasion. However, we're finding that they are heavily weathered. But we're just so gratified and blessed to have been there and to participate. And we did collect some excellent specimens, Triceratops and Edmontosaurus. And remember, this is the Lance Formation. So now we can conduct a comparative analysis of our findings of Triceratops and Edmontosaurus from the Hell Creek with those that were found at the Lance Formation. And we're hoping to make this a regular part of our dig schedule. And the thing that impresses us the most is the way that they engage so many students on this dig and the way their students attend secular meetings around the country. We think that's wonderful. That's something our students do. And so we're very excited to be associated with all the Hansen Ranch folks. And we thank the Bulwarks and all the other folks who are involved at that ranch. It's just a wonderful opportunity. And we're so excited to work with Southwestern Adventist University and Southern Adventist University. We're hoping that their students will also come into this work full time. A lot of them do come to the digs over the summer, but there's a lot of future opportunity for students in the Adventist system to work with us on these bones. We did find some interesting finds there. We found a tooth. Pectinodon is one organism that is collected there, found there not in great numbers, but Pectinodon teeth have been featured. We found a tooth also, a theropod tooth, and so we're waiting for that to be identified. But again, we're just really excited to be able to go to the Hansen Ranch dig in Wyoming, and we're looking forward to a long relationship with them. The new facilities at the ranch are simply spectacular, and we appreciate the leadership of Art Chadwick on this project. Our second dig this spring and summer was in Montana again at Glendive in the Hell Creek. We spoke earlier of our finding of nematodes after Kirsten Peterson's project. We dug many soil samples. One nematode was found and collected from soil samples. One nematode was also collected from the fruit trap method, which we discussed earlier. But we actually, because of the poor results we were getting from the soil and from the fruit, we decided to extract a freshly dug bone shard from the location where the Triceratops horn was located and where other bones had been collected from that site. We put that bone shard into a Bowerman funnel and waited a few hours, and we actually collected a worm that came out of that bone. And here's a nematode that was collected from a bone shard. This is another world first discovery. It's a preliminary discovery. We had dissecting microscopes with us at the dig to check our work with the Bowerman funnels, but we had no attachment camera, so we were able to use this cell phone to videotape through the eyepiece, and you can clearly see this moving worm. This could have only come from the bone, which was thoroughly cleaned before it was placed in the funnel. And so this is a world first discovery, a living organism, a living nematode inside a dinosaur bone. This was later identified by an expert at Virginia Tech as a fungal eating nematode, which is what we expect because we found so many fungal infestations deep within these bones. So this is a world first. It does need to be corroborated. More specimens need to be captured and sent to the experts in Virginia for identification, but we're very excited about this world first discovery. We need to corroborate this over and over again, which we intend to try to do. But after we filmed this phone cell video, I was able to collect that nematode from our specimen container, put it into fixative, and transported it back to the DSTRI lab, where we were able to photograph it under high magnification using optical staining methods like Nomarsky, as you see here. And this was identified by a scientist, a PhD scientist at Virginia Tech. He identified it as from the Apellicaida, I guess that's the way it's pronounced, Apellicaida, and that's a fungiferous or fungus-eating nematode, which is what we expected based on our discovery of fungal fibers, fungal hyphae growing throughout these bones, particularly in Montosaurus. And so we're very excited about this identification of a fungiferous worm, a fungus-eating worm, and so much more work has to be done on this. But this is another world first that we're really excited about, and we're hoping to capitalize on this as well. We're also finding very interestingly tiny insect remains, microscopic jaws, and also bodies, complete bodies, and other insect parts in our decalcification solutions. And so we know that insects are entering these bones, even tiny ones are getting in there and predating upon the soft tissue and fungus and bacteria that are living inside of these bones. And so that's another world first. We can't find any examples of insect parts in the literature in dinosaur bones. There are quite a few papers on the activities of insects and other organisms on these bones and the way they leave the remains with burrowing tunnels and jaw marks and things like that. But we've never seen anything in the literature that shows insect jaws and full bodies of insects. So we think that's another world first. There's also one thing that's being discussed by a lot of folks out there, this idea that red blood cells are being found in these bones. That's not the case. Most of the images that are shown as putative red blood cells are much larger. Red blood cells average about 7 microns in diameter, and most of these round circular red structures, which we now know as framboids, are much larger than that. We have learned quite a bit about these framboids, which people are calling red blood cells, from our epifluorescent microscope studies in the UV using thin sections of these bones. We're able to look in the canals, we're able to see if there's vessels or not. These framboids are found within vessels, they're all jammed together, we're calling them clots. These framboids are also found in canals where vessels are gone or absent, and they're not uniform in shape and size. They differ in size, they differ in shape, they differ in reactivity to the UV. Also in our thin sections, we're finding these clots that push out of these vessel canals and into surrounding bone. And these framboids are present there also, all jammed together, all connected together as if they were clotted. And so I don't think there's a way that we can associate these structures that we're seeing today in these iron oxide framboids and what the oval nucleated red blood cells would look like in dinosaur bones if we could see them. Models would be avian or reptilian models for comparison, but they're never the right shape. They vary in size all over the place. They vary in reactivity, and we have actually done elemental analysis on these clots which contain these framboids, and it's almost pure iron oxide. In fact, one research at the Microscopy Society of America called them iron-rich veins. And so we think this could be the remains of the dinosaur blood. We don't know whether the hemoglobin is in there. Some folks are claiming hemoglobin is found. But we think these are the oxidized remnants and crystallized remnants of the clots that formed when these animals died. We also want to review our collection of an Edmontosaurus skeleton, a complete skeleton, at Glendive, Montana. This was named Ruth. This specimen of Edmontosaurus, a nearly complete specimen, because it was discovered by Ruth Terry Armitage in 21, I believe it was, 21 or 22. The entire skeleton was unearthed and prepared for removal, and we dug it down to a point where the Glendive Dinosaur Museum could come in and able to take it and add it to their collection. We were very proud to unearth that completely, and that should be a great specimen to go visit at the Glendive Museum. But you're going to notice some holes in it because we took specimens for soft tissue study. We took a piece of the jaw, beautiful jaw that was collected from that specimen. We also took two pieces vertically displaced from each other from the sacrum. The thinking is a lot of times that these dinosaurs simply fell over and died as a natural death, in which case there would have been livor mortis, which is the pooling of blood to the decumbent or lower portion of the body. And so we took specimens from the sacrum that were about 20 millimeters or more separated from each other, and so we should see evidence of blood pooling in these bones, in the clots that we're studying, if in fact they did fall over and suffered through livor mortis. We suspect that that's not the case. But we also took a beautiful vertebra from the Hell Creek this summer, and so we have a lot of work to do on Edmontosaurus and Triceratops from Hell Creek, and we're hoping to do a wonderful comparative analysis of the Triceratops collected in Hell Creek and whatever differences might exist between Triceratops collected in Wyoming, and the same goes for Edmontosaurus. So we now have the same species from two different formations, and we're looking forward to doing a collaborative study on that. So if you go to the museum and look at Ruth in the Glendive Fossil Museum and you see chunks missing, that's courtesy of DSTRI.org, because we're studying those for soft tissues and clots. Now, we love to have volunteers on our dig sites. Last year in 2023 we had something like 15 volunteers. In 2024 we had 8 to 10 volunteers, and so we'd love for you to come and help us. We don't charge. In fact, we're all volunteers. We do invite you to come on our dig free of cost. The only costs that you will bear are a $100 a day fee to the dig operator, sometimes less. If we get a large enough group of volunteers, we can negotiate that price, and then you're just living expenses. So some organizations are charging $1,500. We don't do that. You can come on our digs for free, and we welcome your help. We need your help, and so we are shamelessly begging for helpers to come this year. We're going to dig in Jordan, Montana in 25. This is where we've collected Nanotyrannus before, so we're going back to Jordan. That's the very center of the Hell Creek Formation, and so we invite you to come along with us. That'll probably be in the early spring. Then we hope to go back to Wyoming, to the Hanson Ranch in June, and also we have a trip planned to Oklahoma. We're going to visit the Oklahoma Museum of Natural History, the Sam Noble Museum there, and they've given us opportunity to collect from their museum specimens. And this is Permian, and we've already published several papers on the Permian. Some of the nerves that we found in the Permian are just so beautiful as compared to Cretaceous. So if you would like to join us on any of these digs, please let us know. You can contact us at DSTRI.org. Section 5 is about the Microscopy Society of America annual meeting, Microscopy and Microanalysis. We were very excited to attend the Microscopy Society of America meeting this year in Cleveland in 2024. We talked about it a little bit previously, but I cannot recommend joining this society enough. For our students and research colleagues, it's paramount, and if you would like a career in microscopy, it's very inexpensive as a student to join. To come to the meetings, it's inexpensive for students. There's travel award money available, but It's just such a powerful meeting, and it's just so approachable, and there's something for everybody. There are pre-meeting congresses, short courses, and specialized courses in teaching available with certificates available. And once you earn these certificates, it makes you more employable in Microscopy Society of America jobs. There's also pre-meeting student activities. So the student union is very active in the Microscopy Society of America, and so before the actual meeting begins, students gather to do specialized courses for them and instruction and training. And so it's really a neat opportunity for students to come and get engaged. There are lots of opportunities for socialization. The Society works hard to create meeting space and opportunities for students to meet, for focused interest groups to meet, for the local area societies to meet. And we were able to connect with several of the Western local area societies during this year's meeting. And we hope to be able to attend several. And there's one in Arizona, there's one in Texas, and of course in Portland. And there is a society in Washington. It hasn't been meeting for quite some time, but we hope to be involved in re-energizing that group. So all of this takes place at the microscopy and microanalysis meeting. DSTRI students actually get to participate in board level meetings and leadership meetings. We were very excited to have Jonas, our intern and student in El Paso, Texas, attend not only the editors meeting, which discussed the journal and all the happenings with the journal and direction, future opportunities, leadership positions that are open. And they were very, very happy to have him. He also attended the editors' socialization meeting, which was really great. And he met the director of the Royal Microscopy Society, who was here from London, and they actually invited him to come to England, and they offered to pay his expenses to come and speak for him and his family. So there's really incredible opportunities there that you only get when you come to some of these meetings. The meeting tracks during the microscopy and microanalysis meeting, are varied. There are biological tracks, there are physical tracks for material science, there are forensic tracks sometimes. In the past, there have been art restoration and just so many different applications for microscopy that are covered at a professional level with scientific presentations in these different focused interest groups. So there are plenary speakers who are very well known and who run amazing microscopy programs around the country. There's so much fun and entertaining and interesting to listen to. You learn a lot listening to some of these plenary speakers. One of the great attractions is to go into the exhibit hall where many vendors from around the world come and present their products for microscopy, whether it's specimen collection or specimen processing or imaging. A lot of the microscope vendors were there. We got to go to the Zeiss booth. We got to go to the Leica booth. And Leica produced a video, which we'll talk about in a second, for us on a nerve slide that we brought with us. But we spent a lot of time at the Hitachi booths. There were two different booths that we went to. The STEM booth is really an outreach program that Hitachi puts on with their tabletop electron microscope to train students. Students can come and get certification on these tabletop microscopes, and then they're employable. You know, for a summer project, they can actually get a job running this electron microscope in an academic or industrial facility. So there's just so many neat opportunities. And we did spend some time looking at several different things under that tabletop microscope. It was a lot of fun for Aria Cruz, Jonas's little sister, who learned how to run the microscope in about 10 seconds flat. I think she's all of 12 years old and was running around with this microscope, just doing an amazing job. She just picked up a leaf and brought it in from the outside, and the images from that were amazing. So it's really a lot of fun. It really excites young students about microscopy and a future in science. But we also put that microscope through its paces with some of our thin section slides that we make of these bones. And we're always finding new discoveries when we examine these things under these professional instruments. So that was a lot of fun. Jonas had a great time working that instrument also. But then we went to the large Hitachi booth where the full-scale floor stand electron microscopes were, and we were able to do some really neat work imaging some of the nematodes and some of the clots that we're looking at in these thin sections. So it's really a neat opportunity as a student to come and see these instruments in operation, to talk to the vendors, to get suggestions and hints from them, and just to see the possibilities that you could bring into your lab. So we really cannot recommend highly enough the opportunity to come to Microscopy and Microanalysis. 2025, the meeting will be in Salt Lake City, Utah. And we intend to present and also participate in board level meetings. And so if you can, register as a student, join the society as a student. I think it's $50 to join. Just go to microscopy.org. You can also join the student activities, give them your email, and you'll get all kinds of emails and announcements and posts from them. And then when the time comes in the early spring, go ahead and register to come to the meeting. You can register as a student and book your hotel. There's reduced rates for hotels right there at the convention center, and there's restaurants and stuff nearby. But again, there's so much to do, so much to learn. It's so exciting. You can interact with other students, look at all the posters that are there, and learn a lot in many different areas of science just by coming to the Microscopy Society of America meeting. So we hope that you'll join us there in 2025. When we went to the Microscopy Society of America meeting this summer in Cleveland, we took along a type nerve, this Cretaceous nerve that shows what nerves should look like. You can see this beautiful crosshatch pattern all up and down. We had put this in osmium for quite some time, and it produced a fascinating result. You can see this line of lipid droplets that was squeezed out of the nerve when we put it on the slide. But that changed over time. Every time we photographed it, after a few days, those lipid droplets coalesced together, and they made larger blebs as time went on, and eventually it turned into one bubble that moved completely across the slide, and you can see remnants of it in the lower left corner here. So the lipids are fascinating to us, and we used another novel way at the microscopy meeting to look at these lipids and show their shape and presence along the nerve fiber. When we attended the Microscopy Society of America meeting, we went to the Leica booth and we used their confocal microscope, which is a laser-based microscope. It uses unique wavelengths to point at your specimen. Now you can see the thinness of the specimen because it was compressed when we put it under the slide pressure, and that means it was malleable before we even put it on a slide. But you can also see the crosshatch very clearly on both sides of this nerve. And also you can see color differences, and these are a result of the lasers that are used. They have a piece of software that can determine the density of different materials in the nerve. And so we asked them, can you strip away the collagen and show us just the nerve blobs inside of the nerve? And so with their software, they were able to peel away the density of the collagen, which shows up because of the lasers, stripping that all away and showing those lipid droplets present along the whole length of this nerve. So this is a very exciting finding. In section 6, we'd like to just take a few moments to describe what the Dinosaur Soft Tissue Research Institute can do for your family, and particularly for your student. We have some wonderful opportunities that we'd like to describe. You may have a student who loves dinosaurs. Many young students do really go crazy about dinosaurs, and that might be enough to attract them into science. You might have a student who's already really excited about science and wants a career, but you don't know how to go about it. How do you get them into a program that would benefit them? What kind of careers are available? Well, in microscopy, the opportunities are literally stunning, and right now is a great time to take advantage of them. So what does DSTRI offer for you and your students? We have several pathways that we can talk about, but I think the most important thing that we need to stress is we literally come for free most of the time. We only charge students a small fee under $70 for our 3-hour hands-on lab, which we'll talk about in a second. But essentially all of our activities, all of our training, all of our mentorship, all of our guidance and opportunity to get into this field and have a wonderful career here is free. We do this because we want to give back. As I mentioned before, I've had a wonderful career in microscopy, and I just can't believe how many jobs there are right now. I wish I'd have had this opportunity when I was the age of some of these 14, 13, 12-year-old students who really want to get into science and get going with it. But all of our publications are free. Everything we publish, we give away, and we bring fresh bones everywhere we go from our digs. These bones are not rock, they're still bone. We love students to handle them, pick them up, smell them, feel them. We've even had students beg to lick the bones, and we say, well, go ahead, that's on you. But they're so excited to be able to handle real dinosaur bones, teeth, and some of these specimens that are just so amazing. And so we bring all that for free with us. But by way of an introduction to microscopy, we have a couple of pathways that students can take. First of all, we come for free to do presentations. So we've often gone into schools. I think there's 8 or 10 schools, public and private, that we have gone in and we've taught 7 or 8 periods in a row. And so the teachers know that we're coming. They prepare their classroom for the extra size because the classrooms usually get packed out when we come. And they just cycle their students through, their AP students, biology students, and any other students who are interested in this topic. And we really literally teach for 7 hours straight with a little lunch break. And that's one way that students can enter this pathway and learn at their own school what we do and what opportunities are available. We simply present our published works. A lot of folks teach to the controversy because, let's face it, dinosaur soft tissue is quite controversial. But we don't do that. We don't teach the controversy. We only teach our published science. And so that's the initial pathway that your student could take advantage of. Talk to your science teacher and have them contact us at DSTRI.org, or you can email president@DSTRI.org, and we'll arrange to come in and teach, as I just mentioned, at your school. And so you can do that as an initiation to the pathway. The other very interesting pathway that we have, the hands-on 3-hour microscope labs that we bring in. We bring 20 brand new, beautiful college microscopes with us, fiber optic illuminators to light everything up. We have hundreds and hundreds of specimens that we've produced over the years that students get to handle. We bring the bones that they can handle. We bring thin sections that they can look at under the microscope and handle. And so for 3 hours, they get a primer, really, on dinosaurs, the soft tissue that is in their bones, and they get to actually handle the bones and specimens and see things like nematodes in the bone that we've talked about, see the clots in the bones, things like this. And so that does carry a cost. We do charge $65 to the student for that. We strongly suggest that students do chores, do extra work around the neighborhood, mow some lawns, wash some cars, walk dogs, and earn that money, because then that student comes in the classroom and they have skin in the game. They've already put in the effort to get the money for that tuition, and they will be blown away. It's really remarkable to see how students change from when they come in the class until after they leave with all that hands-on time. We actually bring bone juice with us, where we have decalcified, soaked these bones in a weak acid, and they kind of fall apart. And that has the cells and the nerves and the vessels inside of it. And we make bone juice slides for them to take back to their microscope in the hands-on labs, and they get to examine those, and they find cells, they find nerves by themselves right away. So it's really a lot of fun to do a hands-on laboratory. We do require something like 60 to 80 students because we pack up the van with 20 microscopes and all kinds of extra gear, and we drive that across the country. We've been to Minneapolis, we've been to State College, Pennsylvania. We've been to Cleveland, Ohio. We've been to Texas. We've been all over the country with this van, our beautiful little blue van. And so we'd love to come to your school and set this up. And so if you can, Mom, if you are interested in this, you can contact us. We have a workbook that we can send you that helps you plan out and set up one of these labs. And we come for free. The only charge is for the students to attend. And it's really the cost of a babysitter, a pizza, and a movie. And they get to start looking at a career that they could jump into in science once they finish their college experience. So we think that's a really neat opportunity. And we've offered this, I think, in 39 different locations around the country. And it's been very successful. Do please contact if you'd like to put on a hands-on microscope lab at your school. Another pathway that we have, and we often do this after we do a hands-on laboratory at a school, is to do an intensive. We do a weekend intensive at your school. We'll bring in 6 to 8 microscopes, enough for 5 to 6 students to come and sit at. And we work all day Saturday, and Sunday, if possible, we'll work in the afternoon. But it's really kind of a stress test. We put the students through a lot of hands-on work where they're working with pipettes, they're working with bone juice, they're working with live nematodes and other organisms that they can handle and examine under the microscope. And so they get a good 6 to 8 hours, maybe even more if we work on a Sunday afternoon, of instruction and training. And wow, the excitement that builds after that is amazing. We've had students tell us, I have to do this work. And so then we commit to mentor those students and to go to their school on a regular basis for hands-on training and mentoring. Not only that, we provide workstations for free. We bring in about $4,000 worth of equipment in a complete workstation with 2 microscopes, maybe a camera. You have to supply your own computer. But that way the students can take pictures, they can email us their pictures and show us their progress, and lots of glassware that they use, the pipettes and the dishes and slides and cover slips to start doing this work. We make a contract with the school to install this workstation for free, and students can go in there and do their work. And we will supply them the dinosaur bones and mentoring and instruction, and they begin to make their own slides and they begin to take their own pictures and learn how to collect these tissues out of the bones. But that's where our heart is. That's what we want to do in order to introduce these students to science and provide them a pathway. So that's a pathway for research that they get to enter, and then they start collecting results. We teach them how to collect their results, document their results, and actually write up papers and presentations. And then, because of the local area societies and the microscopy and microanalysis meeting every year, students can come and present. They can do posters, they can do platform presentations, and of course our intern Jonas Cruz has already done that. And we have other students in the pipeline who in 2025 will present at the meeting. So you have a pathway for research, you have a pathway for presentation and publication. The other pathway that we provide during that process is a contact pathway where you're now contacting other students, other mentors, possible professors which run a program that you could matriculate in and get your academic training under someone like that. So we just can't find a better pathway than this for introduction, for hands-on training, for intensive work, for research completion and publication, and a launch into a career that is just fabulous. And these are some of the career opportunities that have come across my email just over the last 4 or 5 weeks. Every week I get 5 or 6 brand new job openings, announcements, and so there are constant hirings going on. And a lot of these jobs go unfilled because there's no one to fill them. So in terms of career, if you're a 20-year-old, 25-year-old, or whatever, I'd say even up to 40 years of age, if you want to get a pathway into this field, even at 35, 40 years of age, you can do that. And you can probably have a 25, 30-year career ahead of you. at $80,000 a year or more. So we would like to help you get into that career. If you're serious and you're honest and you really want to pursue this and you're willing to do the work, we will help launch you into this kind of career.